Welcome, ladies and gentlemen, to the video on sex linkage. We are going to be talking about traits that are located on the X chromosome. We are going to start in the world of chromosomal theory, which is just stating some points about stuff that we kind of already know about. For example, we understand that chromosomes carry genes. They are the units of heredity. They are the things you pass from you down to your children. We also know from the process of meiosis that homologous chromosomes meet in prophase 1 and then line up on the metaphase plate in metaphase 1 and then separate in anaphase 1. We understand that that has to happen in order to reduce everything down to gametes carrying half the genes, or being haploid or N. Chromosomes also, as we understand it, sort independently, meaning that they line up along the metaphase plate in any rhyme or reason with their partner, and the way that they separate out is very random and by chance. What was happening when this idea of chromosomal theory is developing? There was this man named Thomas Hunt Morgan, and he was an American geneticist. He worked with my least favorite insect in the whole world, a fruit fly. And why did he work with it is because he was trying to understand linkage of genes based on gender. So Drosophila melanogaster, or the fruit fly. The fruit fly is a very useful little insect to use in genetic experiments because of a couple things. It has a very rapid life cycle. It lays lots and lots of eggs at once. You can cross them and have multiple breeding times at any time in their life cycle. They mature very quickly, and the key part here is that males and females are different, meaning they have very different markings, and they only have very few chromosomes. Therefore, it's really easy to understand what chromosomes are linked to what traits because there's so little of them. If you look at the picture provided, you will notice that the female is much larger than the male. Their bottom half of the thorax of their body is longer with different striping pattern than the male's, which is shorter with a large black bottom. The other thing that is prevalent is that you will notice the other picture is the red and white eye. Now, red and white eyes are controlled by a dominant and recessive allele, and Morgan was looking through and trying to determine how that was passed. For chromosomes in fruit flies, let's look specifically at their eye color. We know normally we would say let big R equal red and let little r equal white. But we are going to assume this time that the X chromosome plays a role in this. We do know that's true, but we're pretending that we didn't, and we're going to try it out and see what happens. Our new legend, because of this, has to look like this. We still are using our dominant and recessive letters, big R and little r. However, because we're saying that they are located on the X chromosome, we have to also put the X in front. The X lets us know it is sex-linked and it is found on the X chromosome. We don't change the dominant recessive letters. We still leave big R and little r, but now we just attach it to the X chromosome. We're going to do two practice crosses to kind of look at how this works. We are given the cross of a female fruit fly with red eyes crossing with a male fruit fly with white eyes. How do I know? X big R gives me red eyes. X little r gives me white eyes. This is no different than a monohybrid cross. We are going to find out our parents, create our gametes. So there's the female gametes. Both had dominant R's with their X's. And here's our male gametes. Now you'll notice though, there's something different in the case of sex linkage compared to monohybrids. And that is the males have a Y chromosome. We know this because XX determines female, XY is determinant of male. When you are doing your crosses, if it is talking about sex linkage with males, you have to include the Y in that process. We're going to do the Punnett square, no different than we did before. So bringing together these two gametes. The egg with the sperm will give us this option. This egg with this sperm will give us this option. This egg, with this sperm gives us this option, and this egg with this sperm gives us this option. The difference, however, when it comes to sex linkage is when we are doing the ratios or the possibilities, or what we would call probability, of having red eyes and white eyes changes a little bit. All these offspring have red eyes. We can see that because they all have a dominant allele. But we also have to recognize that these offspring are of different genders. There's males and females. So when we make our phenotypic and genotypic ratios, we have to separate males and females. In this case, the females are both heterozygous, X big R, X little r. So they get this too. There's two of them. Oh, I covered it up by accident. We also know that the males are both dominant, both X with big R's. So there's two of them. And we reduce it down to a 1-to-1 ratio. We can see red eyes, but we have to remember we have to separate it out for male to female. The genotypes is the same. The genotypes are the same. Heterozygous female, dominant male. So we do a 1-to-1 ratio, separating it out because of males and females. We're going to take a female from this cross and a male from this cross, and we're going to use them in our next Punnett square. So our female is now a heterozygous red-eyed fruit fly, and our male is going to be a dominant red-eyed fruit fly. Again, you're going to find out there's no heterozygous for males. You either got red eyes or white eyes because there's only one X. So I'm going to make my gametes again, this time separating out my dominant allele and my recessive allele. And then I have my dominant allele with my X and then my Y, because again, there's a male chromosome here. I'm going to do my cross, and I'm going to end up with these. Now, if I look at it, I have 3 of them that are red-eyed and 1 that is white-eyed, but I can't put that as my phenotypic ratio because 2 out of the 3 are female. I have to separate out female to male. So what I'm going to do this time is the one lone red-eyed male, X big R Y, is going to get the 1 on my phenotype. My 2 females both have red eyes, so they get the second part out of 2. And I have 1 white-eyed male. He gets the 1. So my phenotype for this cross is a 1-to-2-to-1, separating out males and females. My genotype, on the other hand, is all different. They all look different. The female is both dominant, homozygous dominant, then it's heterozygous. My male is a dominant and a recessive. They all have different looking genotypes, so they get a 1-to-1-to-1-to-1. Morgan concluded that genes controlled eye color must be found on the X chromosome, and that the Y gene doesn't carry anything for eye color. Males also couldn't be heterozygous for sex-linked traits because they only have one X. I keep saying sex-linked traits, so let's just quickly talk about what we're talking about here. Sex-linked traits are almost always found on the X chromosome, hence why we use the X's and the Y's to show that they're linked to the X chromosome. Y chromosomes is usually for sex determination in males, so if you get a Y chromosome, you'll be male. There are Y-linked traits, but there's very few. X-linked traits occur more in males than females, and Y-linked traits only occur in males. Y-linked traits make sense that they only occur in males because only a male will pass a Y to another male. The reason, though, why males only the X-linked traits occur more in males than females is because males have only one X chromosome. They only need one dominant or one recessive allele to be affected. So a male can get a recessive trait from mom, and that would then be present in their phenotype. And we'll see examples of that shortly. Again, females cannot get Y-linked traits because they only have one X chromosome. So here is a practice problem that we can go through to look at how to do sex-linked problems. There is no difference to these problems than there would be a monohybrid. The only difference is you have to remember your X's and Y's because it's sex-linked. Sex or X-linked. We're going to cross a red-eyed female fruit fly who is heterozygous with a white-eyed male fruit fly. Legend. Start with your legend. I need to say that X big R is going to be my red eyes and X little r is going to be my white eyes. I'm now going to make the parents. I have a heterozygous female and a recessive male, reminding yourself to include the X's. So my female has to be X big R, X little r, because that is showing heterozygous. X little r, Y would be the male who is recessive. It doesn't have two X's because he's male, he has to have the Y. Make your gametes, female to male. Here is my female gametes. Here is my male gametes. Again, proceed through the cross like normal. And here are my possibilities of individuals that could be created. Again, possibilities. Reminding ourselves that when we're doing phenotypes and genotypes in sex linkage, we have to separate out males and females. So I'm looking at my females and I see there's a 1-to-1 ratio for eye color, 1 red, 1 white. I will also see that in my males, 1 red, 1 white. So my phenotype is a 1-to-1-to-1-to-1, separating males and females from each other. I will also notice that heterozygous is the first individual compared to a recessive, homozygous recessive individual for females. Again, a 1-to-1 ratio. For males, it's a dominant and recessive trait. So again, they all get separate numbers in the ratio, a 1-to-1-to-1-to-1. I want to just cover a couple examples of sex linkage in humans that you may have heard of. The first one is color blindness. We know that color blindness happens because the cones in our eyes don't work properly, but what you may not have known is that this is actually located on the X chromosome. So when you inherit it, you inherit the X chromosome with a functioning pair of cones, or it could be faulty. Now, when we look at how we would define this, X big C is normal, X little c is colorblind. Again, reminding ourselves the X's are there because this is sex linked. It's found on the X chromosome. The other thing I want to point out is you'll notice a colorblind individual is XC with a dash under the C. That just represents that it's a lowercase letter. Some letters in the alphabet don't look different when you write them in uppercase and lowercase. This is just to show that that is a recessive little c. So the phenotypes for females, you can have a normal female that is both normal, dominant, homozygous dominant, 2 big C's, or they can be heterozygous, or what we call a carrier. They show normal, but they have that recessive allele. You can also have a female who is colorblind by having both recessive alleles. This is where you will see that males either have it or don't. There's no in between. When a male inherits a good copy of the gene, X big C, they're just normal. Why? Because they have a Y chromosome. There's not a second X to play in a role here. But if they inherit a bad copy of that gene, the lowercase c, then they become colorblind. There is no carrier or no heterozygous individual here because they have the Y chromosome. Another example of sex linkage in humans is hemophilia. Now, for those of you that are not familiar with hemophilia, it's all about the trouble of clotting blood in your body. You lack the factors that help you clot or reinforce the platelet plug. You may remember this from Bio 20. And essentially, the platelets go to the surface and then that fibrinogen wraps around it. We are lacking that in a person with hemophilia. So if they bump themselves or cut themselves, their blood does not clot. This is actually found on the X chromosome, and more boys are affected than girls, for the same reason that we saw before. They either have it or they don't. The phenotypes for females can be homozygous dominant normal, a heterozygous or carrier individual, or they could have hemophilia with X little h. Males, on the other hand, either have it or they don't. They either have the normal gene or they have the hemophilia gene. Hemophilia, like I said, means you don't clot. So if you bump or hurt yourself, you will bleed internally, which causes swelling and bruising and all that stuff. Here are some pretty famous people that had hemophilia. Hemophilia actually is quite common in the royalty lines because people married within their families. They didn't go outside of their families to marry. So that causes an issue. You pass that gene down over and over and over. Here's just a little funny tidbit of a fruit fly. Thought it was kind of funny. Now, hemophilia, you can see from that picture, is just the swelling and bleeding around joints normally. You'll see a picture there with Miss Edmondson. She's wearing the Canada toque. there is a friend of hers that his name is Jeremy, and he is going to be available to speak to you guys Tuesday in a Zoom link with Miss Edmondson. He has hemophilia, and so can answer some of the questions. He's going to talk about it, and he is very interesting in the sense that nobody in his family has hemophilia. So Miss Edmondson and him will discuss kind of how that happened. I just want to quickly touch on a last couple few things about gender traits that are kind of stuff that is outside of what we're learning with sex linkage. But essentially, you have sex-limited and sex-influenced genes, where sex-limited means it's expressed in only one gender, or sex-influenced means it's more common in one gender. So milk production in cows is expressed in female cows. Bulls carry the gene, but they don't actually make the milk. Where male pattern baldness is influenced by the testosterone that's in the body. Females have testosterone. They need the recessive genes to make it happen, but females can suffer from baldness. It is not as common in females as it is in males. So that's just an example of how sex linkage can actually be broken into a variety of different topics. We're not going to get into these. This is just showing you the differences. We're also going to not talk a ton about, but you will see Y-linked traits. Essentially, Y-linked traits, ladies and gentlemen, are located on the Y chromosome and are passed from father to son. Again, females don't have a Y, so it's not passed within females. An example of a Y-linked trait is actually male infertility, which is, well, as we learned from before, can be a very common thing in a couple's life. Another interesting thing that has an influence on sex genes or X-related chromosome diseases is Barr bodies. Now, Barr bodies are essentially women have two X chromosomes, therefore we have an extra chromosome than men. They call this gene dosage, meaning we have double the amount that we need. Obviously, men function with one, but women have two. So we actually don't need both to express both of our genes. So that's where Barr bodies come into place. What happens is throughout your body, one of your X chromosomes becomes turned off and is called the Barr body. Cells that have same X chromosome inactivated tend to group together and form patches. Now, Barr bodies are kind of hard to explain without showing you, so I'm going to link a video in about Barr bodies and identical twins that explains them in a very good way. Please watch that video. Calico cats are a really interesting example of Barr bodies. So normally, your hair color for the cat is determined on the X chromosome. The allele for orange is O, and the allele for black is B. Now, understanding right now, guys, that you're probably like, why is there two letters? This is something we'll learn later on. It's called incomplete dominance or codominance, but we don't know this right now. So just roll with it that O means orange and B means black. So for males, that means if they have XO, they will be an orange cat. And if they have XB, they will be a black cat. For females, they need both copies. XOXO means orange and XBXB means black. But we know females can be carriers. They can be a combination of both X and O. X and O and B. XOXB. So what does that mean? Well, it means that throughout their entire body, some of their X chromosomes become inactivated and cause this patchiness. So the patches happen when you have the orange phenotype showing, XB is inactivated, or the black phenotype showing when XO is inactivated. And we call these calico cats. They're a patchy mix of all the colors. Now, the question is, do male calico cats exist? If you think back to the chart that you saw, XOY gives you orange, XBY gives you black, you would say, no, that's not possible. There's no mix, there's no heterozygous individual for a male. But the interesting thing is you can have a male calico cat. But unfortunately, not only is it calico, but it has a rare disease in cats called Klinefelter syndrome, which you may remember from last chapter. Klinefelter syndrome is XXY. So they present male, they are a male cat. However, they have two X chromosomes. In that case, they can have an XO and an XB and have the same process happen with Barr bodies, but are a male cat. I am going to post another video on probability and run through some examples with you. Please watch this one next, and I hope you have a great day.